Boa tarde a todos. É com enorme prazer que nós vamos começar um simpósio hoje sobre doenças autoinflamatórias a respeito de uma nova entidade reconhecida recentemente. Autoinflammatory Disease is a newly recognized entity. Este simpósio foi preparado pelo acadêmico Jorge Kalil, que eu imediatamente quero chamar à mesa, assim como o nosso presidente Rubens Belfort. Por favor. Como você quiser. Não há problema. E eu gostaria imediatamente de passar a coordenação para o acadêmico Jorge Kalil, que dirá algumas palavras sobre por que ele resolveu organizar este simpósio, e a seguir nós teremos os nossos convidados, sendo que o acadêmico Kalil será o primeiro a falar. Por favor, acadêmico.
Muito obrigado, senhora presidente, acadêmica Eliete Bouskela, meu amigo ex-presidente Rubens Belfort. É um prazer muito grande nós organizarmos esse evento. Esse evento, então, agora falando em português, antes de a gente tornar para falar em inglês, nós resolvemos organizar porque essa doença, chamada doenças autoinflamatórias, elas são muito mal conhecidas. Na verdade, provavelmente, nenhum dos senhores aprendeu nada sobre isso nos bancos da faculdade. E mesmo recentemente, a gente sabe relativamente pouco. E vocês verão, pela apresentação de todos os nossos convidados estrangeiros e o convidado nacional, que houve um progresso enorme nesta área, e a gente começa a reconhecer ou identificar vários problemas médicos que não eram identificados como sendo doenças autoinflamatórias e que não são doenças autoimunes. Eu vou tentar mostrar para vocês no início da sessão. É diferente de doenças autoimunes, apesar de também ter esse componente inflamatório importante, porque ela toca uma outra área da imunidade. E por que nós achamos que era importante? Então, é importante porque ela teve um papel super interessante nas diferentes pandemias que houve na história da humanidade, inclusive agora na Covid. É importante porque nós temos que identificar essa doença, porque cada vez mais nós temos tratamentos específicos e há vários tratamentos que estão sendo desenvolvidos no mundo como um todo e que podem tirar o paciente de um grande sofrimento com um medicamento relativamente simples, mas, como eu disse, por não ser identificadas, por não termos o hábito de reconhecê-las, muitas vezes os pacientes sofrem e morrem antes de qualquer tipo de tratamento. Então, eu achei que é uma novidade para todos aqui na academia e resolvemos, então, fazer. Nós estamos fazendo um esforço muito grande de trazermos todos os médicos brasileiros que têm contato com esse tipo de paciente, para que eles também sejam, digamos, de certa forma, informados sobre como reconhecer a doença e como tratar essa doença, para que os pacientes não morram. Então, fundamentalmente é isso. Eu digo, é algo novo e nós organizamos isso muito com a ajuda do meu assistente, que vai ser o último a falar, que está aqui, é o Dr. Leonardo Mendonça. Trabalha lá conosco, dentro da área de biologia clínica, propriamente. Então, começamos já a nossa apresentação um pouquinho mais cedo. Então, vamos começar. Vou colocar o primeiro slide mais alto. Mais alto? Então tá. Tá bom assim? Ótimo. Bom, então, we should switch to English because there are many of the speakers that listen to us. And so what I'm going to say from now on will be in English. If there is any doubt, just let me know. So I'm going to talk a very, it's an oversight on the history and future of these autoinflammatory diseases and put this in the context of the medical knowledge that we have nowadays and how the capacity of identifying genes that we had now because of the everything that is related to DNA sequencing, identification of genes could help us in identifying the disease. First of all, I'd like to remind you that the immune response has two arms that are very important. One, the ancient, can you see it? Yeah. Can you see if I point here? No. Okay. Perhaps here it's better. So the immune response, it starts with what we call a very ancient system of defense called the innate immunity. And the innate immunity, you have some barriers that are natural barriers, like the epithelial barriers. And then when a infectious agent enter the epithelial barrier, they will find the phagocytes, the dendritic cells, NK cells, complement, and that are not specific for that particular microbial, but rather they are recognized by what they call the DAMPs and PAMPs that recognize structures that are not present normally in the human cell. And from this, they trigger a defense that is non-specific. And in this defense, we have many molecules that are synthesized and secretized. And those are, perhaps the most important one is the interferon, but we have TNF and other molecules that are present. And in many cases, the innate immunity is enough to completely eradicate and completely eliminate the infectious agent. If it's not, and these take hours, but there is a connection between the innate immunity and what we call the adaptive immunity. And I'm sure that adaptive immunity, you have heard a lot, and it's involved in many issues, and this is concerning the specific immune response. And the and the connection between those two is mainly due to the dendritic cells, macrophages, etc., that are going to present the foreign antigen to the immune system for both the T lymphocytes and B lymphocytes that will create a specific antibodies in the case of the B lymphocytes after maturation, production of plasma cells and production of antibodies, and also T lymphocytes that can be effector TCD8 cells or CD4 cells. So there is an interplay between innate and adaptive immunity, and I would say that in the 20th century, we have studied a lot the adaptive immunity, and we didn't pay attention at all on the first arm of the response, that is the innate immunity. But yes, it is important, and it's very ancient. So if you take from the history, when you take the Odyssey, Ulysses had met Scylla in the region of South Italy, and Scylla was a woman that was considered like a monster because she could be very kind, and all of a sudden she would become very angry, a lot of fever, and should be bad humor, and etc. And further, later on, when we revise what has been, I mean, all the symptoms that she felt, we knew that actually she was the precursor, what we much later on, we called the familial fever of the Mediterranean. So you're going to speak a lot on the familial fever of Mediterranean. That was also very important during the plague, because the people that could survive in the plague were the ones that had this disease. So this disease, as I put in my in the video that I invited you to this meeting, these people that had this abnormal secretion of interferon, that's the Mediterranean familial fever, they could be able to avoid or to destroy the Yersinia that was responsible for this plague. And we'll see that also it had a very important role in COVID-19. So a disease that can help some people to avoid a particular other disease. So in the beginning of the 19th century, we had this Nobel Prize from Paul Ehrlich that identified the antibody production, and he also identified what we call the oral autotoxicus. So he described that if you inject an animal with blood from another animal, from another species, this animal will become sick because it will reject the blood that has been injected, and would create a storm, nowadays, you know, of cytokines, and create a whole bunch of medical problems. But this is autotoxicus, and this is always we think on the autoimmunity. But more recently, almost in the 20th century, Dan Kastner from USA and Isabelle Tuyutu from France, they described what was called the oral autoinflammatories. That means the autoinflammation causing damage. And there are two sets of diseases that seem to be very important that I use here as an example. One, this is autosomal and dominant, and this is the case of this disease called TRAPS, where there's a defect in the receptor of TNF. And the other disease is recessive, and this is the familial Mediterranean fever, where we have an inappropriate production of interferon. So if you take here and compare the concept of autoinflammatory diseases with autoimmunity, I would like to remind you that autoimmunity is you have self-reactive T cells or self-reactive B cells. It's specific for a particular antigen or a specific organ, and always they are associated with one HLA antigen, like rheumatoid arthritis, like many other spondylitis, ankylosis, or scleroderma, whatever. They are all linked to a particular susceptibility is linked to a particular HLA antigen, and this is caused and triggered by the presentation of this antigen by those genes, HLA genes, that trigger autoimmunity. And how it triggers is still subject of controversies. Myself, we have demonstrated in rheumatic heart disease that this is due to the recognition of the cross-reactivity between a part of the M protein and proteins from the myosin from the heart. You have T cells that recognize both and trigger disease and cause damage to the valves. Others have other explanations for how we trigger autoimmunity. But in autoimmunity, we have specific antibodies or specific T cells. On the other hand, on the autoinflammation, you have the recognition of a particular infectious agent by the pumps and dumps those are receptors that recognize either DNA, RNA, or proteins, parts of the proteins that normally are not present in human beings, and they either start producing large amounts of interferon for the interferon pathways, or they can cause the inflammasome, a specific response that will eventually cause also the inflammation with a cytokine storm that is not specific for a particular antigen and not even for a particular agent, infectious agent, could be any. What is interesting is that we know that autoimmunity, you have genetic susceptibility, and this is mainly due to HLA molecules, as I mentioned to you. Autoinflammation, in most of the cases, as you'll see, I'm going to present you some of the data, but it will be, people will go much more in detail, the ones that are going to present after me. These are a genetic-based disease. Even though it's a genetic-based disease, it can appear at any age. So you can have the gene that's not expressed and may be expressed at any age. And you can find those diseases that are genetic determined very late in the life of the people. And this can be due to either mutations at the genetic level from the early beginning, or can be mutations that will happen in somatic cells, in a particular set of somatic cells. So that's why they may appear late in the life of someone, and it's sometimes very difficult to identify the gene because only that particular set of cells have that particular mutation that bring to the disease. But anyway, when you have undefined or undiagnosed diseases where you have periodic fever and systemic inflammation, you have to think in autoinflammatory disease and go after the genes or go after the ways of identifying it. So if you take in this concept of autoinflammatory diseases, and you take from the beginning of the century, there were 1 or 2 genes that have been identified. But nowadays, with all the tools that you have to do sequence the genes and see the disease, you can see here the number of genes that have been identified. So far, 555 genes have been identified. So this represents 155 different types of diseases that sometimes they have the presentation, the syndrome is similar, but the genes that is related could be different. If you only see for instance, you know that IOS, International Union of Immunological Societies, has a committee that overlook all these publications and the description of the diseases. And it used to be called the Immunodeficiency Committee. Now it's called Inherited Errors of Immunity, because most of them are inherited, completely inherited, or a mutation. You can see from 2022 to 2024, the numbers of genes that have been added to these diseases. So more and more genes are being described, I would say, every month with a particular patient, with a particular new variant or new mutant. So if you take the case of the plague pandemic, the plague pandemic, the problem is related to the production of interleukin-1B, 1 beta. So normally, when you have the contact with your senior pestis that causes the plague, you have the production of interferon and many other cytokines. And pyrene is the one that neutralizes and do not leave the caspase 1 to be activated to produce high amounts of IL-1 beta, just enough quantity to defend the organism. But if there is a mutation, that is the case of the people with the familial Mediterranean fever, then the caspase 1 is activated, and there is a large production of IL-1 beta that leads to pyroptosis and death of the cells. So this is an ancient mutation that happened in the Mediterranean region, but people could survive from the plague pandemic because of the capacity they had of production of interferon and IL-1 beta. This is a Bruegel painting where he shows how he saw the plague that, that killed 40% of the population of the world. And this mutation on the Mediterranean fever happened probably something like 3,000 years ago in the Mediterranean region. And there are 2 mutations that have been described. And by the migrations of the population, they were distributed in different areas. So you can find patients with this Mediterranean fever in different areas of the world because of the migration, but they are due to this mutation in the period. So if you take the case now of COVID-19, there are many patients that had problems in the production of interferon. Some of them producing too much or too little. And this could cause more or less inflammation, and many of them died. So if you take the genes of the post-interferon receptor signaling, for example, like JAK and STAT and STAT2, those that had mutations in these genes had a poor prognosis and a much worse disease. So here we have again, the interplay between an infectious agent and the predisposition of people that have different mutations that sometimes help them to survive to a particular disease, but in many cases predispose them to die. There is a work very interesting from the group from Casanova from France, where he has shown that 10% of the patients that die of COVID, they have antibodies against interferon-1. The elderly people, sometimes they do make antibodies against interferon-1, and this is a major cause of non-response against COVID and then die. So we have these diseases, the autoinflammatory diseases, but do we have a possibility of treating disease? I would say that there are two types of disease. One is much more related to interferon and all the cascade of cytokines produced by the interferon, one, and the other that is linked to inflammasome. So what can we do? Well, very recently, there are many new drugs that appear in the market that can interfere with the action of interleukin-1 beta, that you know that is the main cause of fever. One is anakinra that blocks the receptor of interleukin-1 beta. The other also competes with interleukin-1 beta, then it's the canakinumab, monoclonal antibody that inhibits. And there is the rilanocept that blocks the different cytokines and hamper those cytokines to make their action. And in this case, they can be very beneficial to patients. Things get more complicated in the other areas, in the activation done by interferon alpha, that all the signaling in the interior of the cells, because this is related to the receptors, JAK1, STAT1, STAT2, and others, but also there are many new drugs that are the JAK inhibitors that seem to be very important in blocking this inflammation and have been largely been used in human disease. And if you want to go further in the capacity of treating those patients, we're going to listen to people, that we'll present today on how we can use CRISPR-Cas9 based gene editing to cure, for instance, IPEX. IPEX is due to a mutation in FOXP3. FOXP3 is the most important molecule to trigger regulatory cells. So the cells that regulate the immune system are, FOXP3 is by far the most important, and the mutated FOXP3 give a syndrome called IPEX, that you have diseases all over because you do not control your immune system, and you have a lot of autoimmunity. And there is a possibility of treating those patients by correcting the FOXP3 by using human stem cells and treating those cells, correcting by CRISPR-Cas9 the defect, and then re-inject this in the patients. The same thing also we can do, we can think in treating the stem cells in the DADA2 problem, also a mutation that have this syndromic effect of autoinflammation. So there is a whole world that's being developed. For doing this, we do need to identify the genes, the mechanism of action, and interfere in the mechanism of action. This is very modern medicine. Instead of trying different medicines by chance, you can identify really the genes, how the genes can work in the different cascades to cause disease and interfere in this disease by a particular molecule. So that's what we're going to hear today with the different speakers, and I'd like to thank you for listening to me. Thank you. So in continuation, we are going to listen now to monogenic vasculitis, when we are going to suspect of such a disease. And this will be done by Dr. Marco Gattorno, one of the leaders in autoimmunity, autoinflammation, from the University of Gênova, Instituto Giannina, Gaslini, in Italy. So, Dr. Gattorno, thank you very much for being with us, and let's listen to your talk. Mhm.
No. Thank you, Leonardo, and thank you to Professor Kalil for the very kind invitation. Unfortunately, I cannot be there, even live, because when you have the meeting, I will be in Helsinki for the press meeting. But however, it's a great pleasure to give this talk. We will talk about monogenic autoinflammatory disease, but in this specific, the monogenic vasculitis. Also is a great topic, I know, also for Leonardo, and we share this very interesting a lot of interest on this topic. These are my disclosure. We are talking about vasculitis. This is a very interesting topic. These are the classical classification for the multifactorial vasculitis. And you know, here I'm a pediatrician, and these are the most frequent vasculitis that we observe in childhood. What I would like to take here in our conversation is the presence of many monogenic mimickers of vasculitis. We will talk about the pediatric ones. I will not talk about Vexax because Leonardo is much more experienced than me, but I think it's very important to understand that there are some vasculitis that are monogenic, and their number is really growing. We have a clear idea of what polyarteritis nodosa is, which is the clinical features of this manifestation, the fact that the patient have subcutaneous nodules and livedo racemosa, and they have the classical histological manifestation with a fibrinoid necrosis of the walls of the medial or small arteries. That's why we have to go in deep for the biopsy. As you know, no glomerulonephritis, no pulmonary involvement, no ANCA positivity in polyarteritis nodosa. There are plenty of different indication or recommendation for polyarteritis nodosas. I think it's important because they're taking care about methotrexate, azothioprine, and other cyclophosphamide and other treatment. And I will tell you how important it is to recognize the monogenic form, because the treatment is completely different from what is recommended from the typical polygenic multifactorial polyarteritis nodosa. I'm talking to you about the so-called adenosine deaminase 2 deficiency. It's a disease that we know since almost 10 years now, and is an important disease because it gives rise of the first example of a monogenic disease that is caused by a single gene mutation, at least two mutations, because they are autosomic recessive disease of this gene that is ADA2. As you can see here, in comparison with the previous slides of multifactorial polyarteritis nodosa, the clinical features are completely the same, especially for the vascular manifestation. You can see the nodules, you can see the livedo racemosa, you can see the ischemic features, and also the same histological features. This is really relevant. Of course, what is very peculiar of this manifestation of this disease is the presence of neurological manifestation. They are mainly related to stroke and other clinical features that you can see here in my slide. This is quite important. It's quite special and peculiar of this condition. In this condition, there is a huge endothelial activation with inflammation and the presence of inflammatory parameters like TNF and interleukin-1 alpha. As you know, ADA2 is an homology, has an homology with ADA1, is a mostly extracellular, and seems to have a very important effect of growth factor. In patient, we have normal level of ADA1, but very low level of ADA2 concentration in the plasma. And there are, of course, many different mechanisms, possible mechanisms related to this condition, also related to a possible activation and role, intracellular intracellular or of other one, have a sensor of DNA. But the most important part is related to an overactivation of the netosis. ADA2 reduce the ADA-induced net formation. The neutrophils of the patient with ADA2 are rich, very rich of neutrophils, and the netosis that is really high, related to the defect of this enzyme that is able to cut the netosis. And there is an idea that the increased netosis is able to activate the macrophage to produce huge number of pro-inflammatory cytokines. First of all, TNF. There are plenty of studies of netosis, but of course I don't have the time. And this lecture is more related to the clinical features of this condition and other monogenic disease. Some years ago, as soon as we have the first cases, we try to have a survey in Italy on this condition. We also set up an enzymatic test that is very important. It's very important to have an enzymatic test. At that time was a very complicated enzymatic test in which we test the presence of adenosine deaminase 2 with HPLC method. We need the fresh monocytes from this patient. We stimulate with adenosine, and then we look to the presence of the derivatives of adenosine, inosine, and epoxyxanthine after the use of this inhibitor of adenosine 1. So, due to this collection of patients, it was clear that the clinical manifestations were quite various. You can see the vascular manifestation of our Italian patient at that time, some years ago. The neurological features, that will come back with the neurological feature, especially the strokes, hemorrhagic and ischemic stroke. Some patients have a sudden hearing loss. That is a typical manifestation of ADA2, very severe, because it's a sudden and complete even loss related to the vascular involvement. And some patients have also hypogammaglobulinemia. As far as the neuroimaging findings is concerned, some time ago we have a study on Italian study on with our neuro radiologist with 12 ADA2 patients that go into details to analyze all the images of ADA2 patients, and they found out a lot of radiological signs in this condition: intracranial hemorrhagic manifestation, as you can see here, acute brain ischemic infarcts, and also chronic brain ischemic infarcts. So even hemorrhagic and ischemic infarcts. There are some strange perivascular enhancing tissue you can see here in this patient. And what is quite interesting, that this enhancing tissue disappear after the proper treatment. We will talk about on that. We have some patient with posterior encephalopathy that was reversible. And then also patients with intracranial aneurysm and chronic brain and medullary ischemic infarcts. So the variety of possible clinical findings related to vascular damage in brain is so important, is so relevant, and sometimes could be very dangerous for the patient. What was quite interesting, since some of the patients have hypoglobulinemia, our friends in Germany, in Freiburg, looked to the patient with antibody deficiency in their cohort some years ago, and they test for adenosine deaminase 2, and they find out that a relevant number, 11 out 181, was actually also was positive for ADA2, a genetic mutation. They have also vascular manifestation and the same clinical features that I show you. So sometimes the antibody deficiency can be the most relevant part of the treatments. Some of the patients can have infection, but most of them do not have a very important infection. Otherwise, some of them needs IG replacement. And I would like to talk to you about these two twins, because this is a very important part related to this condition. There are two twins that we follow many years ago, and now are followed by San Raffaele Hospital in Milan. And they have livedo reticularis, neutropenia, hypogamma uremia. They have ischemic stroke. There are two very important parts related to these two twins. The first part is related that you can see here that both of them were heterozygous. It was very strange. They were heterozygous like the father, but the father was completely asymptomatic, while the two twins were both very symptomatic. This is quite interesting, and that's why I told you that it's so important to have an enzymatic testing at home, because we were able to test that indeed the patient 1 and patient 2, the two twins, had a very severe decrease of completely absence of ADA2 activity, while the father was completely normal, as you can see here. That is the first point, important point from this fever. Sometimes you cannot get a mutation, two mutation, because sometimes they have deletion that can be missed with the normal next generation sequencing. And so you have to do another different approach with MPLA to identify deletion. This is an important point. If you have heterozygous, theoretically should be normal. If you test for enzymatic test and the enzymatic test is completely pathologic, you have to ask to your geneticist to look for the second mutation. This is quite important. But the other second message from this family is related to the fact that they have hypogammaglobulinemia, but they have also some very severe hematological manifestation. Neutropenia, severe neutropenia, and bone marrow failure for the second twin that go into bone marrow transplantation some years ago with a very good outcome. So this is the enlarging spectrum of ADA2. We understood at a certain point that beside the vascular involvement, vasculitis and ischemic strokes and so on, some of the patients can have also bone marrow failure, hematological manifestation, with red cell aplasia, thrombocytopenia, pancytopenia. And this is quite relevant. These patients are really severe and sometimes, as I told you, have to be transplanted. It's clear that patients with missense mutation tend to have a milder form with more vasculitis. Rather, those patients that have more severe variation, like deletions or duplication or non-sensitive variant, are more prone to develop also the hematological manifestation, because there is a decrease of residual ADA2 activity. This is quite important. Some other clues that came from the study of the number of patients that has been described using the Eurofever registry. The Dutch community collected the Dutch patient with ADA2, 29 patients. Was very interesting. Also, they collect also some adults. One quite worrisome aspect is that the high rate of malignancies in this patient, 8 patients out of the 29 developed malignancies, and especially 5 with hematological manifestation, especially T cells, large granular lymphocyte leukemia. And this is something that we have to take in account very carefully, the possibility to progress towards hematological tumors. This is a very important message from this patient. Another important patient that sometimes maybe we have missed some patient. We are talking about the recognition of this monogenic vasculitis. And the first point that I always told about this point is the story of this little girl of 15 months that have the classical phenotype of ADA2, fever, livedo reticularis, myocarditis, strokes, hypogammaglobulinemia. She responded well to anti-TNF treatment. We will talk about that. And she had no ADA2 mutation. And again, the enzymatic test was absolutely positive. So it was pathologic. So we were sure that was something missing. That was confirmed at that time also by the laboratory of Mike Archfield at the Duke. We also look to the whole exome sequence of the trio, just to understand if there was another gene that was around that could be explained this phenotype and also the functional defect. But then when we make the whole genome sequencing, we understood that the patients have a homozygous duplication. So not deletion, but duplication of a very big part of the gene that it was inherited by the two parents that was heterozygous. And this huge duplication, of course, make a very big breakpoints that have a deleterious effect on the protein. So this is another message. Sometimes the patient can be either negative, not only heterozygous, but seems to be negative. And sometimes we have to really look with the more sophisticated genetic analysis to have the final diagnosis. So that's why it's so important to have the enzymatic test. Why is this so frequent in ADA2? Because you have to understand and to remember that the gene of ADA2 is indeed in the part of chromosome 22q11 that, as you know, from the DiGeorge syndrome, is subjected to a number of copy number variation. And so in this part of the genome is quite frequent that you can have copy number variation, so deletion or duplication. Another missing patient, I always told the story of Luigi that had a very tremendous story. He was an adult, 42 years old, and you can see here patient with recurrent fever, persistent elevation, acute phase reactant, rhino phenomenon. Then he have neurological hearing loss, cutaneous vasculitis with severe leg ulcers, ischemic stroke, bowel ischemia, hypertension. It's a very bad and incredible severe condition and disease. You see here the treatment that Luigi had during his very long history: oral cyclophosphamide, azathioprine, rituximab, methotrexate, Dapsone. None of this condition was able to help him. At a certain point, our friends in Florence suspected maybe Luigi has the mutation that Marco always tells to us, and indeed that was the case. The enzymatic activity was pathologic, and he has 2 mutations. So this is very important because we think that there are several adult patients that present with a story of polyarteritis nodosa and that are unrecognized. So if you have patient, if you are an adult expert and have a patient with this kind of history, very early onset or familiar manifestation of vasculitis, think about ADA2, send the enzymatic activity to Leonardo, whatever, to us, and if there is positive or pathological, send for genetic analysis. And this is quite important because after the very interesting study from Pouilly in Boston, it's clear that the carriers of deleterious ADA2 variant is quite high, 1 out of 262 patient individuals. That means that the disease prevalence is 1 out of 2,200,000 individuals. That means that we would expect a very relevant number of patients worldwide, 35,000 patients. That means something very similar to FMF. So, as you know, FMF is the most frequent disease as an autoinflammatory disease, and for sure, ADA2 seems to be, together with FMF, the most important and most frequent disease. We calculated that in Italy, in which we have the 56 million of inhabitants, we calculate that we could expect 250 patients. So you know the number of the people that you have in Brazil. I don't know, I think that 130 or 20 millions, I think. So it's more or less the double. So we can expect 500 patients in Brazil. So you have to look to these patients, to ask to adult rheumatology following vasculitis, to talk a lot with hematologists that now tend to be very careful on this condition, very interested also. In this Italian grant that we received for the health ministry, that we perform with our colleague of San Raffaele Institute in Milan. There was several very interesting aims. One of them was to set up an enzymatic test and to test and to validate. This is Alessia Cafaro in our institute that set up this test that is very easy. You can perform on Ristopot, send just the paper. And now we are collecting a number of patients and normal individual and carriers. And we found out this test very, very sensitive and specific for the patient that have very, very low level of ADA2. And so you can make very early diagnosis in patients very, very quickly. The other part of the grant is related to understand a little bit more the hematological features. It's quite interesting. This is the paper, the first paper from the group of San Raffaele with all the patients, also from Genoa and San Raffaele, in which we make the bone marrow aspiration. We make bone marrow aspiration either in patients with hematological manifestation and those that do not have hematological manifestation. And I have to tell you that even those patients that do not have hematological manifestation, the non-severe, gray, have a clear defect. Most of them have a clear defect in the colony formation. That means this is intrinsic to the genetic defect. In Genoa, we are, with our hematologist, we are studying how to correct in the bone marrow this defect of colony formation. And I think this is a very important point because, as you understand, this is the most severe manifestation of ADA2. ADA2 has to be recognized very quickly because as soon as you have the diagnosis and even the suspect with the pathological enzymatic activity, you have to start anti-TNF treatment because indeed anti-TNF treatment is the treatment of choice for this condition. You can see here the dramatic effect on vasculitis and the other dramatic effect you can see here, the stroke in our study before the treatment and after the treatment. These 17 patients that we have in NIH, they have less patient, 15, but older patient with a huge amount of strokes, 55 during their life. And you can see again the number of strokes after anti-TNF initiation. So you don't have to be a statistician to understand that this is indeed anti-TNF treatment is so important because prevent the occurrence of neurological manifestation. Of course, in those patients that have severe hematological features, anti-TNF treatment doesn't work, and hematopoietic cells transplantation is the only cure. And this is a survey some years ago on from Isabelle Métier in all over the world. There is some space for enzymatic replacement. As you know, there is one enzymatic replacement. Maybe there is some role also for ADA2, pegylate ADA2. We will see in the future. Of course, there is a great hope also for the possibility of gene therapy. We are studying with our patient, our colleague in San Raffaele, Milan, on the possibility to treat also ADA2 patients, not only ADA1 patient, as you know, in San Raffaele, in other few centers, the gene therapy is a possibility. Okay, so we are talking about the medium vessel vasculitis, polyarteritis nodosa, but there are other conditions that can mimic a vasculitis in autoinflammatory disease. And this can mimic an ANCA-associated small vessel vasculitis. These vasculitis are extremely, extremely rare in pediatrics. We very few cases of microscopic polyangiitis or granulomatosis with polyangiitis in pediatric age. But there are some patients that can have a similar condition This is the story of Martha with skin lesion, facial papers, with pustule and scars. You can see here. Then she blames. She had ANA positivity, but also ANCA positivity. She also had lung manifestation with severe restriction and focal thickening and interstitial lung disease, very severe. She has a SAVI syndrome. This is quite interesting because SAVI, as you know, is a part of interferonopathy. This patient can develop some skin manifestation and features related to that can mimic a multifactorial autoimmune disease. There are some reports of some patients with ANCA associated vasculitis that was really related to SAVI. And you can see also in different members, because it's an autosomical dominant disease, that presented the same feature of ANCA vasculitis, was labeled as an ANCA vasculitis, but had indeed a SAVI. This is quite relevant because, again, there is a different treatment in this condition with JAK inhibitors and now with anifrolumab. That is different from, of course, what we use in ANCA positive vasculitis. Why these patients have autoantibodies? You know that there was a paradigm that say that in autoinflammatory disease, autoantibodies should not be present. Indeed, this is different in interferonopathies because interferon has a great role in activating autoreactive B cells that we have in our body. And so that's why in a situation in which there is a huge presence of interferon alpha, you can have autoantibodies that sometimes can be pathogenic. Okay, and I told you that anifrolumab, of course, could be a very good drug. With anifrolumab, we are able to completely make a negative interferon signature in this patient that was never achieved in patient with JAK inhibitors. So I think this is a very promising drug for this condition, sometimes in combination with JAK inhibitors. Okay, so as you know, the number of autoinflammatory disease is expanding, and some of these conditions also can present with vasculitis. There are different conditions. There are some conditions that can mimic a multifactorial other mimickers of vasculitis. As you know, A20 haploinsufficiency can relate it to a Bachetta-like manifestation, very, very similar to Bachetta. They are related to overactivation of an NF-κB pathway. There is another condition that is a relay haploinsufficiency here. And you can see here also this patient can have pustule, apoptosis, and ulcers that can mimic again Bachetta. So there are some monogenic conditions that can mimic another vasculitis. This is an even more recent disease, deficiency of L4, that have the same clinical findings that can really mimic a Bachetta condition. That's why we have to need for the monogenic condition. And the last point is related to some other syndrome. Very recently, some patient with this perinatal onset neutrophilic cutaneous small vessel vasculitis, ANCA negative, was detected. They have also liver fibrosis, 2 of them. And this was related to the novel truncating missense variant of SRC family tyrant kinase, LIN. There is an overactivation due to this severe mutation of this potent pro-inflammatory manifestation, pro-inflammatory. And this is quite interesting because these patients are related, and this syndrome are related to mutation of protein kinases. The protein kinases are, as you know, enzymes that are very important in many biological activity with phosphorylation of amino acids that are involved in major cellular process, differentiation, proliferation, and cell death. There are many genes, more than 500, and 2% of human genomes are related to protein kinase. And that is very interesting because there are some other conditions related to these variants of SRC family kinase. For example, this condition related to another kinases, that is HCK, that are again linked to vasculitis and to severe, for example, here, pulmonary involvement. So this is quite intriguing because as the tyrant kinase are so important, as you know, in many immunological features, but also in the activation of the endothelium, and SRC family are related to integrate. So that's why in some of these conditions that we are trying to understand, they are related to kinase defect. We found a lot of patients with vascular inflammatory manifestation, vasculitis, as a presentation. And in this new group of conditions that we call kinasopathy, related to the defect of tyrosine kinase. So I finish here. I hope to convince you that beside the classical multifactorial vasculitis, there is a growing number of monogenic vasculitis that we have to understand and to test with genetic analysis. And I think this sometimes is very important because they can really lead us to make the diagnosis, a correct diagnosis, but in sometimes the most important things also to start with a proper treatment that sometimes is completely different from the treatment that we would use for the multifactorial disease of which these conditions are mimickers. So that's why it's important to think about, to make the genetic analysis and the functional enzymatic test as soon as possible, because it's very important to start treatment very soon. So with that, I would like to thank you again for the kind attention, to wish to you a fantastic meeting and a fantastic time in Brazil, I hope. And to that, I would like also to thank all the people in Genoa that works with me on this topic since many years. Thank you very much indeed.
Bem, nós estamos com um problema. O Dr. Gattorno está em Helsinki e ele gravou, por sorte, nós estávamos com medo, e ele gravou a aula dele para entrar só agora na discussão. Só que está instável, né? A ligação dele conosco, ele não está conseguindo uma ligação firme conosco. E então vai dificultar muito as perguntas para ele. É uma pena, porque ele tem uma pessoa que tem uma cultura muito interessante. Mas ele, pelo menos, nós tomamos a precaução de gravar, porque a gente estava com medo que ele estando fora de casa, no hotel onde fosse, ele pudesse ter problema. Mas, em todo caso, senhora presidente, acho que nós podíamos estar abertos para... Vai ficar gravado. A conferência vai ficar gravada. Eu sei que para muitos de vocês apareceu aí uma sopa de letras de difícil compreensão. Mas eu acho que é super importante a mensagem que ele nos deixa, que às vezes síndromes que a gente vê, vasculite, por exemplo, você pode ver clinicamente de diversas formas. E se utiliza normalmente os seus procedimentos padrão de tratamento e diagnóstico. E por trás tem muitos desses doentes que têm defeitos genéticos que nós não identificamos. E que esses pacientes, então, eles são subtratados, maltratados, ou não são tratados, e nós perdemos esses pacientes. E por isso, o esforço muito grande que nós temos feito de que a gente aprofunde no diagnóstico de muito. Nós estamos trazendo aqui uma pequena parte das doenças autoinflamatórias, mas a gente tem que aprofundar no diagnóstico, porque a gente pode efetivamente tirar o paciente de uma situação muito ruim às vezes de uma maneira muito fácil, o tratamento específico, se a gente identificar que gene está envolvido ou que mecanismo está envolvido. Alguma pergunta?
Kalil, só antes, eu não posso fazer pergunta para ele, para o apresentador de antes de você? Ele não vai ouvir, né?
Acho que ele não vai ouvir,
fazer...
mas faça a pergunta de qualquer jeito, nós vamos mandar para ele e vamos responder.
First of all, I would like to congratulate you both for the remarkable presentations. I am very happy. I take part to the Brazilian group that works for the immunomediated inflammatory diseases. And now we have just published the Brazilian guidelines to prevention of this disease in terms of infectious diseases. And can I speak in Portuguese? Because, you know, the question will be to you mostly. E é muito interessante essa apresentação que ambos fizeram. Eu queria pedir licença à senhora presidente para fazer um comentário, né, como membro do Grupo Brasileiro. Nós publicamos recentemente, Kalil, as normas e guidelines para prevention de infectious diseases em doenças imunomediadas, inflamatório-imunomediadas. E vocês fizeram, eu teria uma lista de perguntas para fazer, mas vou fazer apenas duas. Quer dizer, primeiro, o quanto essas doenças autoinflamatórias elas são prone, elas são facilmente cooptáveis pelas doenças infecciosas, o que é algo extremamente importante no Brasil, tendo em vista o fato de que vocês usam inibidor de TNF-alfa e, portanto, colocam essas doenças muito facilmente candidatas, digamos, a doenças infecciosas. E a segunda seria qual é a diferença do ADA2, já que a ADA é um marcador inflamatório tão importante nas doenças imunomediadas e em outras granulomatoses que nós tratamos com mais frequência. Essas seriam minhas duas perguntas que eu acho que você mesmo poderia responder, Kalil. Muito obrigada, senhora presidente.
Bom, acho que são muito pertinentes suas perguntas. Por isso, a precisão do diagnóstico, né? Dá um anti-TNF, anti-interleucina, anti-isso, anti-aquilo, em pacientes cuja indicação não é muito precisa, porque a gente sabe que ele tem uma secreção inadequada dessas citocinas, nós podemos causar muito mal. E, por isso, o diagnóstico tem que ser muito preciso, que realmente esse paciente tenha deficiência numa determinada via, uma via de produção de citocinas, ou está hiperativada uma determinada via. E por isso a insistência que a gente está tendo aqui. Né? Você viu que ali a quantidade de poliartrites ou polivasculites, que a gente não, que são de causas que não são as causas que normalmente a gente pensa e que por isso pensaria um outro tipo de tratamento, é muito grande. Eu concordo com você que qualquer tipo de tratamento em que a gente utiliza que causa uma diminuição da imunidade para que a gente trate a doença, a gente tem que ter uma precaução muito grande com a infecção, principalmente num país como o Brasil. Né? Você sabe que se nós pegarmos doenças até mais comuns, que tem muito aumento de L17, L23, etc., doenças autoimunes, e que a gente trata, a gente tem um aumento muito grande de tuberculose, por exemplo, né? Para dar um exemplo que tem. Então, por isso que é uma medicina complexa, é uma medicina refinada. Nós precisamos de centros no Brasil que olhem com muita atenção essas doenças, para que possam seguir esses pacientes. A gente não acredita que qualquer hospital na periferia vai conseguir acompanhar esses pacientes mais complexos, e por isso, digamos, o esforço que nós temos feito de criar centros em todo o país que olhem com muito cuidado essas doenças, em que a gente tem muito intercâmbio, muita gente vai a São Paulo e a gente está constantemente viajando. A gente, que eu digo, são eles, né? Para que a gente possa instruí-los para fazer a distinção, para ver quando é que a gente dá um determinado medicamento ou outro. Vou te dar um outro exemplo de doença infecciosa, justamente. Nós vimos há muitos anos atrás que eu quero até trazer aqui para uma reunião da academia, que o grande problema da doença de Chagas é a secreção inadequada de interferon gama e os indivíduos, a hipertrofia do coração se dá muito frequentemente porque eles têm outros problemas genéticos. É uma coisa parecida com isso aqui. Tem outros problemas genéticos, geralmente na produção de energia. E aí o sujeito hipertrofia o coração e faz a cardiopatia dilatada. Então, nós vamos começar um ensaio clínico para tratar os pacientes com etarnesept, TNF, alfa, etc. Por sorte, nós resolvemos testar isso bem pré-clínicos em animais. O que a gente viu é que todos os pacientes, todos os animais que nós tratamos morreram porque você desequilibrou a resposta imune e os parasitas proliferaram e terminaram matando os nossos animais de experimentação. Então, nós estamos falando aqui de caminhar sobre o fio da navalha. E por isso que eu vejo a importância do diagnóstico preciso, da capacidade que nós teremos que ter de ver essas vias metabólicas e vias de defesa de uma maneira muito precisa. E mesmo assim tem que ser feito um acompanhamento muito de perto desses pacientes para que a gente não perca. Por outro lado, nós temos uma possibilidade real de tratarmos esses pacientes que a gente diria no dia a dia da medicina, que são refratários ao tratamento e que terminam morrendo. Acadêmico.
Senhora presidente acadêmica Eliete, Bouskela, professor Kalil, eu tenho, durante a minha carreira, tratado muitos doentes com doença inflamatória intestinal. E eu tenho visto nesses últimos, sei lá, 10, 12 anos, talvez, um aumento da relação entre a microbiota intestinal, doença inflamatória intestinal e associada a outras doenças, como artrite periférica, psoríase, etc. Inclusive, no segundo semestre do ano passado, foi publicado um artigo interessante, em que mostra uma disbiose que é comum à doença de Crohn, psoríase e artrite periférica. Então, a minha pergunta agora é se está sendo investigado e se existe alguma relação da microbiota ou do microbioma em relação a essas doenças autoimunes.
Autoinflamatórias. Nessas doenças autoimunes, sem dúvida alguma, tem muito estudo. As doenças autoinflamatórias também, eu acho que estão no começo. O número de pacientes é relativamente pequeno, mas eu acredito que a disbiose deve ter um papel importante. Eu não sei se você quer acrescentar alguma coisa, Léo.
Obrigado, professor. Eu encaminhei as perguntas do anti-TNF nos pacientes com ADA2 para o professor Gattorno, e assim que ele responder, eu encaminho de volta. Mas aqui no Brasil, a gente não vê um risco maior de tuberculose nos pacientes com ADA2 tratados com anti-TNF. Então, a gente, assim, é um grupo pequeno, pouco tempo de acompanhamento para poder a gente afirmar que realmente eles não vão ter mais exposição. Mas é um questionamento interessante. A diferença da quantificação, né, da não conversão da ADA2, da ADA total, a ADA2 ela é muito mais expressa na imunidade inata e mais expressa em macrófagos e tem mais um fator pró-inflamatório do que a defesa contra agente infeccioso. Né? Enquanto a ADA total, que é no líquor, no líquido pleural, tem mais ação realmente contra agentes infecciosos. Então, só uma observação, mas depois eu encaminho a resposta dele. Com relação à microbiota e as doenças autoinflamatórias, faz muito sentido, né? Porque realmente é o primeiro reconhecimento de infeccioso por PAMPs e DUMPs em geral, né? E tem casos bastante interessantes da síndrome de Blau, onde o encontro de uma um comensal, na verdade, na pele, parece ser realmente o desencadeador da doença. Então, parece não ser tão estéril assim o comportamento dessas doenças autoinflamatórias, mas talvez de um mundo infeccioso que a gente não dava tão importância da microbiota, né? Mas realmente tem pouco conhecimento, mas provavelmente muito promissor ainda no patogênese das doenças.
Bom, vamos continuar, então, a nossa discussão durante o café e voltamos pontualmente às 6 horas. Obrigada. A Magda